So on behalf of Qiagen, I would like to welcome you to our webinar, giving you some insight into the QIacuity nanoplate dPCR system. Before I start, just a brief disclaimer. What I'm going to present is not meant to be used for the diagnosis, prevention, or treatment of a disease. Our agenda today covers various aspects, including a short overview of the complete system. We then will have a closer look on the starting template, some typical applications, and the underlying calculations for the analyses thereof. I will conclude my presentation with a walkthrough of the analysis software. Now let's start with the QIacuity system. First, we have the QIacuity nanoplates. Both plates come in a standard SBS format. They either have 24 wells with about 26,000 partitions per well, or 96 wells with about 8,500 partitions per well. In the course of this year, we also will launch an additional plate with 24 wells and 8.5K partitions. Second, we have the QIacuity dPCR instruments, which range from a one-plate instrument with 2 detection channels up to an 8-plate instrument with 5 detection channels. Third, we offer 2 PCR kits for either probe-based or EVA-green-based detection. The first one can be used for the amplification and detection of up to 5 different targets within one and the same well. Not mentioned on this slide are the various assay formats which we are going to offer for mutation detection, CNV, and gene expression analyses, and which we will cover in separate presentations. Let's now focus on the starting template. For dPCR experiments, a very diverse plethora of templates can be and will be used. This includes genomic DNA, isolated from blood, tissues, cells, tumors, cell culture, swabs, lavages, and various other sources. A frequently used starting material for mutation detection and quantification is FFPE DNA, which stands for formalin-fixed paraffin-embedded DNA, isolated frequently from tumor tissue. Circulating free DNA and circulating tumor DNA frequently have been used as biomarkers and starting material for prenatal diagnosis by dPCR. Methylation analysis requires the use of bisulfite-treated DNA, a starting material for dPCR, which usually is highly fragmented and also, upon treatment, AT-rich. Also, a mixture of dozens, if not thousands, of different DNAs covered by the term environmental DNA have been used in dPCR experiments. Not to forget, DNA from pathogens, plasmids, and synthetic starting templates. Of course, also RNA, after conversion into cDNA, is an important starting material in dPCR. Gene expression analyses not only include the quantitative determination of messenger RNA molecules, frequently using total RNA, but also the quantification of microRNAs and long non-coding RNAs. dPCR recently has become an interesting alternative to qPCR in the vast field of RNA viruses, such as HIV, influenza, or most recently SARS-CoV-2. Last but not least, and similar to DNA, also artificial RNA molecules such as in vitro transcribed RNA or RNA oligonucleotides have been used as dPCR template. Now, when starting a dPCR experiment, it is worth, when starting a dPCR experiment, it is worth to spend a few thoughts on the template material used. This includes the purity of the template, its integrity in terms of length, but also its integrity in terms of sequence. The purity, or rather the impurity of the starting template, can impair the outcome not only of a qPCR experiment, but also that of a dPCR experiment. And there are several ways how this can occur. Alcohols and salts can change primer and probe annealing properties and reduce the efficiency of amplification. This in turn will reduce the fluorescent signals in the positive partitions and will impede the successful discrimination between positive and negative partitions. Humic acids, frequently found in DNA isolated from soil, will quench the fluorescence of double-stranded DNA binding dyes such as EVA green. Nucleases might still be present in raw cell lysates and easily can degrade RNA and DNA. Urea and phenol might be introduced by quick and dirty sample preps and tend to denature the Taq polymerase. A completely different way of dPCR inhibition can be induced by acidic polysaccharides, which can be found in DNA isolated from feces. These molecules can mimic nucleic acids and will form dead-end complexes with the Taq polymerase. You might have heard previously that dPCR is less prone to inhibition, and this is at least partially true. Since we are looking on the fluorescent signals only after the PCR has been completed, a CQ shift, as frequently seen upon partial inhibition in real-time PCR, will not change our dPCR quantification results. However, when it comes to a complete breakdown of the dPCR by inhibitory substances, then also the dPCR experiment is ruined. Therefore, dPCR can only work optimally if templates of high purity are used. Template RNA and DNA, which is strongly degraded, will show sometimes large discrepancies between the OD quantified DNA amount and number of copies amplified and detected by dPCR. This is due to the fact that not all DNA or RNA fragments do still contain an intact amplicon site. Therefore, larger than expected DNA amounts might be necessary to achieve desired sensitivity, for instance, in mutation detection. We therefore strongly advise to keep amplicons as short as possible, particularly when using strongly degraded samples such as FFPE DNA or cfDNA. Frequently, strong degradation of template DNA or RNA goes hand in hand with reduced sequence integrity due to residual crosslinks. These will prevent strand separation and amplification. In addition, abasic sites can induce unspecific amplification and detection. Therefore, care should be taken that dedicated protocols and kits for recovery of high-quality gDNA from FFPE samples are used. I now would like to direct your attention to some quantitative considerations with special emphasis on genome sizes, application dependency, and the type of signal detection. On this slide, I have listed the genome sizes of various frequently studied organisms, including Homo sapiens. It might be kind of disillusioning to see that the genome size of the mouse is larger than ours, and I don't know whether it gives you some comfort knowing that the Japanese plant Paris japonica has a genome size 50 times larger than ours. Well, why have I listed all this information? This I would like to explain on the following slide. A question which is frequently linked to dPCR: which template amount should I use for my experiment? Given that you even have the choice to decide, the answer is it depends. Imagine you are looking for a single copy gene, which means 1 copy per haploid genome. If you decided to always use 10 nanogram of DNA as template, this would correspond to copy numbers ranging from roughly 3,000 to 2.6 billion copies per reaction. The latter one would result into hopeless overloading of the QIacuity nanoplates. You will see and learn during this presentation that in dPCR, at least a few partitions must be empty, which means negative, in order to apply the calculations for template concentration. On the bottom of the slide, you see some calculations indicating appropriate template amounts of human genomic DNA if studying a single copy gene. When you are looking for the needle in the haystack, meaning a very rare mutation, you rather want to go with a higher template amount. When looking for copy number variation events, a significantly lower template amount will do the job. This slide demonstrates how This slide illustrates how the template amount used affects the outcome of the experiment depending on the detection method. EVA green will bind to any kind of double-stranded DNA and will thereby start to fluoresce. Even if a partition does not contain a DNA molecule coding for the target of interest, it still will contain other DNA molecules. Upon increase of the template, as done here, this background fluorescence will increase and results in worsened discrimination between positive and negative partitions. Therefore, our advice is not to exceed 100 nanogram of template in EVA green based applications. With this slide, I would like to explain why the inclusion of a restriction enzyme is important when using genomic DNA as template. It should be performed whenever you are using DNA samples with an average length of more than 20 kb. Plasmids always should be linearized independent of their size. This ensures even distribution of the template on the nanoplate and is also a prerequisite for the efficient amplification of plasmids, which tend to re-anneal extremely fast as long as they are in a circular form. A restriction enzyme digest is particularly important for copy number variation analyses, where multiple copies of a gene might be linked in tandem. It is not required for highly fragmented DNA such as FFPE DNA, circulating DNA, cDNA, G-blocks, and other short templates. Now, how is the restriction enzyme digest performed in our system? Well, simply include one of the restriction enzymes, which we will recommend, directly in the master mix and incubate after addition of the template for 10 minutes at room temperature. Of course, you should make sure that the restriction enzyme will not cut within your amplicon. And for Qiagen assays targeting genomic DNA, such as CNV assays and mutation detection assays, suitable enzymes for the respective assay will be recommended. With the next set of slides, I would like to give a short review of some typical applications in dPCR and the underlying calculations. In the upcoming month, we will offer some more detailed seminars on those various application types. Okay. Among the numerous applications for digital PCR, a few of the most frequently used ones are listed on this slide. No matter which one you choose, the starting point always will be the absolute quantification of your targets. Based on these results, subsequent analyses such as mutation detection, copy number variation analysis, and gene expression analysis can be performed. What is the outcome of a digital PCR experiment using the QIacuity system? There are primarily two numbers which play an important role for the subsequent analysis. The first one is the number of PCR reaction mix filled, which means valid partitions. Based on the plate type which you have selected, this can be either up to 26,384 or 8,510 partitions. This number is determined based on the number of positive spots per well in the reference channel. The second number is the number of positive partitions per well, which means partitions showing a fluorescence signal clearly above background. In theory, this again can be numbers up to 8.5K and 26K respectively. If performing a multiplex dPCR, those numbers will be determined for each detection channel, which means for each target separately. As I have already pointed out, the starting point of all dPCR analyses is the absolute quantification. I therefore will call it first level analysis. After the analysis and the processing of the raw image data, you will obtain the following numbers: the number of valid partitions per well and the number of positive partitions per well. Please note that only a partition which has been identified as a valid one can be considered as a positive one, given that its fluorescence lies above a certain threshold. Using those two numbers, you automatically can calculate the number of valid but negative partitions. Now, using those numbers, the analysis algorithms will calculate the copies per microliter of your target, including the confidence interval at a 95% confidence level. If replicates have been used, also the mean value of these replicates will be calculated. What are the underlying assumptions and calculations? Well, in a dPCR reaction, the complete reaction is separated into multiple single partitions. The target molecules are randomly distributed across all available partitions. Now, after this distribution process, the partitions will contain either no copy of the target molecule, one copy of the target molecule, or more than one copy of the target molecule. As the target molecules are distributed randomly, Poisson distribution can be used to calculate the average copy of the target molecule per valid partition. Simeon Denis Poisson, a French mathematician, has developed a theory and corresponding equations on the discrete probability distribution of a given number of events, which can be observed within a defined time or, as in our case, space interval. Our space interval will be a single partition. Now, the Poisson distribution itself gives probabilities for positive, integer, random events, in this case, a partition containing at least one copy of our target sequence. Lambda is the expectation value for these events and will tell what the most likely average value of copies per partition is. In order to do these calculations, the software needs the information how many valid and how many positive partitions we see in the well. The equation shown also demonstrates why we always need at least a few negative partitions in dPCR in order to do proper calculations. If each valid partition were a positive partition, then the numerator of this equation would become 0 and no further calculation would be possible. I now would like to cover a few applications which we had been calling second level analyses. All second-level analyses are based on the results of the absolute quantification. Types of second-level analyses include mutation detection, gene expression analysis, copy number variation analysis, and quantification of genome editing events. For mutation detection, copy numbers of the wild type and the mutant target, which are detected on different target channels, are calculated. Then the ratio of mutant versus total number of positive events, including wild type plus mutant, is calculated and expressed in percent as fractional abundance. If replicates have been used, the mean ratio is also calculated and expressed in percentage. Let me explain it with sample number 1. We have detected 2,000 copies per microliter of wild type and 5,000 copies of mutant. They sum up to 7,000 copies per microliter. The fractional abundance of the mutant is 71%. In sample 2, the copies of both targets are much lower. Still, the fractional mutation abundance is identical to sample number 1. And we could go through all of those samples, but this would take quite some time. Therefore, I rather proceed to the next type of second-level analysis, namely gene expression. For gene expression analysis, the copies per microliter of the gene of interest and the reference gene or the reference genes are calculated. The number of reference genes can vary and usually will be in the range of 1 to 3. Within one sample, there can be multiple genes of interest and reference genes, each detected on a different channel. Then the ratio of gene of interest versus reference gene is calculated. If more than one reference gene has been used, the ratio of gene of interest versus the geometric mean of the reference genes is calculated. Subsequently, the ratio of gene of interest versus reference gene of the reference sample is set to 1. The ratio of all other samples are normalized to this value. And let me again explain it with our sample 1. We have detected 2,000 copies per microliter of our gene of interest and found 1,000 copies of our reference gene number 1. From this, we obtain a ratio of 2. Since sample 1 is our reference sample, which we want to compare all our other samples to, we set this ratio to 1. If we now look at our sample 2, the copies of the reference gene is identical to sample number 1. However, our gene of interest has 4,000 copies per microliter in this sample. The ratio now will be 4. And if we compare it to our reference sample, we will learn that our gene of interest is upregulated twofold. The way how we perform copy number variation analysis is quite similar. Copy numbers of the gene of interest and the reference target are calculated. Then the ratio of gene of interest versus reference target is calculated. The ratio of gene of interest versus reference target of the reference sample is then set to the known copy number of this reference sample, in this case 2. The ratios of all other samples are compared to this reference sample, and the corresponding copy numbers automatically will be calculated as it is seen here. With this slide, I would like to briefly discuss two edge cases. When almost all valid partitions are either positive or negative, basically all of them are positive, basically all of them are negative, then the uncertainty of the calculated concentration will clearly increase. The confidence interval easily can exceed 100%, and certain modifications of the algorithms will apply. Now, the sweet spot for minimal confidence interval values lies in the range of 1 to 2 copies on average per partition. But quantification can range from 5 copies per partition on average down even to 1 single copy in the whole reaction. In the last part of my presentation, I would like to walk you through the various analysis modules and displays of the QIacuity software. I would like to demonstrate the various ways how dPCR results can be displayed. In this webinar, I will focus on absolute quantification, since the subsequent analysis modules will be explained in dedicated webinars anyway. And don't forget, absolute quantification is the starting point for all subsequent analyses. In order to analyze a plate, you go to the plate's overview page, search for the plate you want to perform an analysis, and select Analyze from the context menu. You will be encouraged to select the corresponding wells, and you can select whether you would like to perform a channel or a target-based analysis. In this case, we decided to do it based on the target we had been looking for. Of course, you can select also multiple targets or channels simultaneously, and all the results will be displayed in the results window. How will the results be displayed? Well, first of all, there is the list view. It will display a table giving an overview of the wells analyzed. This table will contain the following information: the well identifier like A1, B2, the reaction mix. It contains the icon and the name of the reaction mix. Of course, it will do so only if you have defined it previously. So there's no need to do so, but I can tell you it's quite helpful. Then the next column will contain the target or channel, depending on the settings defined when selecting a source. This column shows then the target or channel names. The sample NTC/control designation. This column shows the sample NTC and control name with its corresponding icon that identifies the sample or indicates whether the entry is a negative control, no template control, or other type of control. The concentration given in copies per microliter. This column shows the concentration assigned to each target or channel per well. If we had identified within one and the same well various targets, because we performed a multiplex experiment, those ones would be all listed in addition. The confidence interval shows the value of the confidence interval at a 95% confidence level. If replicates have been used, and replicates are defined by using the identical reaction mix plus the identical samples, then you also could see the mean value of those replicates. Last but not least, the partitions. This column shows the number of valid, positive, and negative partitions per well and target or channel. On the heat map, you will see the individual quantification results for each sample given in copies per microliter. Below that concentration, you will find the corresponding confidence interval given in percentages. If you activate the partitions view, the number of positive partitions will be displayed. And please note that the number of positive partitions does not automatically correspond to the copies per microliter. And below the number, you will see the number of valid partitions, in this case, clearly above 8,000 of 8,500 available ones. On the histogram view visualizes the fluorescence values of selected wells and the frequencies thereof for the selected target or channel. It has two axes. The x-axis represents the relative fluorescence intensity. The y-axis represents the number of partitions showing that fluorescence intensities. The threshold field shows the threshold value of the fluorescence intensity that is used to distinguish between positive and negative calls. There are two ways to change the threshold. The first one involves pointing on the chart, which triggers the appearance of a dotted line. Once the dotted line is in the appropriate spot, click the chart. The line becomes solid, as we see it here, and the threshold value is updated and displayed in the threshold field. In addition to moving the line, you also can directly edit the value in the threshold box. The 1D scatterplot tab shows 1D scatterplot view for each analyzed target or channel. A 1D scatterplot view again has two axes. The x-axis represents the analyzed partitions, while the y-axis represents the relative fluorescence intensity of each single partition. A red line represents the current threshold fluorescence intensity value that is used to distinguish between positive and negative partitions. Fluorescence values below the threshold are shown in gray, above the threshold in blue color. The threshold field shows the threshold value of the fluorescence intensity that is used to distinguish between positive and negative call. By default, the software will set the threshold automatically. There are two ways to change that preset threshold. The first one, again, involves pointing to the chart, which triggers the appearance of a dotted line. Once the dotted line is in the appropriate spot, click the chart, and the line will become solid and the threshold value is updated and displayed in the threshold field. In addition to moving the line, you also can directly edit the value in the threshold box. To apply the new value on the data, click Recalculate to trigger the recalculation of results. Now this slide depicts the 1D scatterplot display of an EVA green-based experiment where we had been using strongly varying input of genomic DNA. As you've already learned, EVA green-based detection will also detect varying gDNA template background, and therefore it makes perfect sense that the software set individual thresholds for the samples. Now, the concentration diagram shows the diagram that displays the concentration values and confidence intervals for the samples analyzed. One diagram is created again for each selected target or channel. A concentration diagram has two axes. The x-axis shows the analyzed wells, and the y-axis represents the concentration value for the selected targets or channels of each well. The values on the y-axis have two available scales, either linear or logarithmic. Each diagram represents two values for each well. The concentration value, which is displayed as a bar, and the confidence interval displayed as an error bar. To view the mean values for replicates on the diagram, click on Show Mean Values for Replicates. In this case, one bar is displayed for replicate group, showing the mean concentration value and the mean CI value of the replicate group. To view exact values and the list of wells belonging to the replicate group, you can simply point to the associated bar and a pop-up window will appear. The signal map view will display all the partitions of a well, which we see here, which have been identified as positive partitions. Comparing them to the raw image, you occasionally will observe that obvious dust particles, as shown here, and other minute contaminations will be detected automatically by the software suite. Regions where these artifacts can be detected will be excluded from analysis. The QIacuity software suite allows you to create reports of your analysis results. All created reports remain accessible in the software suite and can be downloaded or deleted at any time. It automatically includes run details such as start and end time and software and instrument version. You can include any diagram as long as you don't exceed the number of 24. The report is automatically downloaded in the download folder of the PC as a PDF file. Furthermore, the report is stored in the software suite and accessible via the report environment of the plate. And with this slide, I would like to finish my presentation, and I'm looking forward to taking your questions. So thank you very much for your attention.